Okay, so in the previous lecture, we were discussing the experiment done by Beadle and Tatum in order to study the Neurospora nutritional mutants. These are mutants that have mutations in genes important for the biosynthesis of amino acids. So just a quick reminder about this experiment. Neurospora cells grown on complete medium, and this complete medium contains all the necessary amino acids. These neurospora was treated with X-ray. X-ray will introduce mutations into the neurospora genes. The surviving cells were able to grow on complete media because this is complete media, contains all the necessary nutrients. And then when they took these colonies and grow them on minimal media, which contains only vitamin, salts, and glucose and does not contain any of the amino acids, these colonies did not grow, indicating that these colonies, they are nutritional mutants. Each one of these colonies contains cells that are unable to synthesize a particular type of amino acids. In order to determine which type of amino acids cannot be synthesized by these surviving neurospora colonies, they took each cells from each colony and grow these cells on minimal media plus one of the 19 amino acids. And in this example, the neurospora cells in this colony were unable to grow on minimal media plus valine, minimal media plus lysine, but they were able to grow in minimal media plus arginine. This particular result indicates that the cells in this colony, they are unable to synthesize arginine. That's why they can only grow in the presence of arginine. So these are arginine deficient mutant neurospora cells. So they've done the same thing with the other cells in the other colonies in order to determine which type of amino acids cannot be synthesized by these cells. Of course, the cells in this colony are unable to synthesize arginine because X-rays probably destroyed or damaged or mutated a gene important for the biosynthesis of arginine. Okay. So the conclusion is that a gene important for arginine synthesis is mutated. Okay? And that's why these cells cannot grow on minimal media, only in the presence of arginine. Now, scientists knew that arginine synthesis requires more than one gene and requires more than one enzyme, encoded by more than one enzyme. So in the next step, two scientists, Shab and Horwitz, they wanted to know, or they wanted to determine how many genes are important for the synthesis of arginine, okay, and how many genes could be mutated in order to for these cells to become arginine deficient. Okay. So again, this is the experiment done by Biedel and Tatum, and two scientists, Sub and Horwitz, they continued the work of Biedel and Tatum, and they were able to determine that there are three enzymes, here they are called enzyme A, B, and C, are required for the synthesis of arginine in neurospora. Okay, so this is an example of a metabolic pathway. A metabolic pathway consists of consecutive chemical reactions. So this is a chemical reaction one, let's say. This is chemical reaction 2, and this is chemical reaction 3. So as you can see, 3 chemical reactions, they constitute a metabolic pathway. Now, each one of these chemical reactions is catalyzed by an enzyme, and each one of these enzymes is encoded by one or more genes. Let's call it gene A, gene B, and gene C. So arginine is synthesized from a precursor. This is just madde kham. The precursor is converted into ornithine by enzyme A. Ornithine is converted into citrulline by enzyme B, and citrulline eventually is converted into arginine. So in neurospora, these 3 genes at least are, and their products are important for the biosynthesis of arginine. Okay. Now, so you could think about the cells here, which are neurospora arginine deficient cells. They could have mutations in any of these genes. Because if there is a mutation in this gene, for example, so if a mutation, again, mutation is tafra, destroys this gene will not be able to produce this enzyme, which means that the precursor will not be converted into ornithine, ornithine will not be converted into citrulline, and arginine will not be produced. Okay? This is if the mutation hits or introducing gene A. Also, if a mutation affects gene B and gene B is no longer functional, enzyme B might be absent or non-functional, citrulline will not be produced, ornithine will be produced because gene A is available, is normal. So ornithine will not be converted into citrulline, and citrulline will not be converted into arginine, so there will be no arginine produced. Okay? At the same similarly, if the mutation hits not gene B, but gene C, this means gene C is destroyed or a mutant and can no longer produce enzyme C or produce active enzyme C, which means that citrulline will not be converted into arginine. Citrulline will be found inside cells. Ornithine will be found inside cells because these 2 enzymes are normal and active, because they have active and normal genes. So any mutations affecting any of these genes, or mutations affecting any of these genes, might lead to the absence of arginine from in neurospore. Okay. Based on this information, Sub and Horwitz, they classified the arginine deficient cells into 4 classes, 4 types, and classes and okay. How did they do that? So they took neurospora cells. These neurospora cells are arginine. They are arginine deficient. Arginine deficient means that deficient means ناقص. They cannot synthesize arginine. Then this is the wild type neurospora. This is normal neurospora. So normal spora, okay. So this line indicates growth. So neurospora can normal neurospora, wild type neurospora can grow on minimal media only. These mutants, the mutant arginine deficient neurospora, cannot grow on minimal media. So here we have no growth. Growth means نمو. No growth, no growth. The wild type can grow on minimal media plus ornithine. So this is a minimal media with ornithine added to it. Remember, arginine is synthesized from a precursor into ornithine. Then ornithine is converted into citrulline or citrulline, and then into arginine. So this is a minimal media plus ornithine. This is a minimal media plus citrulline, minimal media plus arginine. The wild type, the normal neurospora, they can grow on all these minimal medias with or without supplement. Supplement means إضافات. These are the supplements. This is one supplement, and one supplement. Okay? Now, when some of the neurospora cells that are arginine deficient were added to minimal media plus ornithine, they were able to grow. Minimal media plus citrulline, they were able to grow. Minimal media plus arginine, they were able to grow. These cells were called class 1 mutants. Okay? These cells, they can grow on ornithine, citrulline, or arginine. Okay? Which means these cells of class 1 mutant, they have a mutation in enzyme A, and enzyme A is not present. If enzyme A is not present, it cannot convert the precursor into ornithine. So, but these mutants, so they are mutants in gene A. So they have a mutation in gene A. But if you give them ornithine, enzyme B and C are good. Which means if you give them ornithine, they will convert ornithine to citrulline, then arginine. That's why they grow here. If you give these class 1 mutants, type 1 mutants, النوع الأول, if you give them minimal media and citrulline, they will grow. Why? Because if you give them citrulline, they have normal enzyme C, they will convert citrulline into arginine. Of course, if you give them arginine, they will be able to grow. Okay? Class 2 mutants, are unable to grow on minimal media, which we already know. If you give them minimal media and ornithine, they will not grow. If you give them minimal media and citrulline, they were able to grow, and they were able to grow on arginine. Notice that all classes of Neurospora arginine deficient mutants, they can grow in the presence of arginine only. On minimal media only, they cannot grow. Okay? But class 2 mutants can only grow in the presence of minimal media and citrulline and in the presence of minimal media of arginine. Why? Because class 2 mutants, it turned out that they have a mutation in gene B. Class 2 mutants, they have mutation in gene B, which means that enzyme B does not have an activity inside neurospore cells. So if you give them ornithine, like here, they will not grow simply because there is no enzyme inside this class of mutants that can change ornithine into citrulline. But if you give them citrulline, they will be able to grow. They are gonna grow because enzyme C and enzyme A are normal, okay? And they are functional, or they are active. That's why if you give them citrulline or arginine, they will be able to grow. The third class of Neurospora arginine deficient mutant. They cannot grow in the presence of ornithine. They cannot grow in the presence of citrulline. They can only grow in the presence of arginine. Why? Because class 3 mutants, they do not have a functional enzyme C. They have functional enzyme B, enzyme A. Even if you give them ornithine and enzyme B converts ornithine into citrulline, enzyme C will not be able to convert any citrulline into arginine. So it doesn't really matter if you give them citrulline or ornithine, because they do not have the enzymatic activity important for the synthesis of arginine from citrulline. Okay. So by doing this experiment, Sharp and Horwitz were able to classify Neurospora arginine deficient mutants into 3 classes. And they were able to discover that there are 3 genes, A, B, and C, that are important for the biosynthesis of arginine. Questions so far? No questions. All right. So this experiment also provided evidence for the hypothesis suggested by Archibald Garrett. Remember, Archibald Garrett, he suggested that one gene encodes one enzyme. And as you can see in this experiment, these 3 genes encode 3 enzymes. But now we know that not all proteins are enzymes. So some genes will be encoding proteins that are not enzymes. Okay? So you have to understand that Archibald Garrett, he suggested this hypothesis to try to explain the relationship between genes and proteins. So Archibald suggested that one gene encodes one enzyme. But now we know that some genes encodes or store the information necessary to synthesize proteins that are not enzymes. So this hypothesis, or this idea suggested by Garrett, was revised, and now it became, and later it became, one gene encodes one protein. Okay? And this one was no longer correct or valid. Later on, scientists discovered that some proteins consist of more than one polypeptide. So remember, you learned about the protein hemoglobin in the Biology 101 course. Okay? So what's the function of this protein, hemoglobin? Do you know? What's the function of the protein called hemoglobin? Hemoglobin بالعربي. Give body color? No. No. It transports oxygen in the blood, through blood. So it's responsible for carrying oxygen from our lungs into all the cells inside our bodies. Hemoglobin consists of 4 polypeptides. Remember when you learned about the 4 levels of protein structure, the primary, secondary, tertiary, and quaternary? Hemoglobin is an example of a protein with a quaternary protein structure. Hemoglobin consists of 2 alpha polypeptides and 2 beta polypeptides. So this protein consists of 4 polypeptides or 4 subunits. 2 alpha, they are the same, and 2 beta. And the gene beta encodes polypeptide beta. Gene alpha encodes polypeptide alpha. So as you can see, to make hemoglobin, the cell requires the expression from 2 genes. So 2 genes are required for the synthesis of this protein. So 1 gene, 1 protein is not entirely correct. It was after the discovery that some proteins, they have quaternary structure and they are made of more than 1 polypeptide. This hypothesis was, or this idea was restarted so that 1 gene encodes 1 polypeptide. Not a whole protein, because this is 1 polypeptide, 2 beta polypeptides, 2 alpha polypeptides, they make 1 protein called hemoglobin. So now, this is no longer valid. This is a more correct basically a more correct statement. Now, also, later on, scientists discovered that for some genes, 1 gene might encode more than one polypeptide. So one gene might encode two or more polypeptides. So some genes, they might encode more than one polypeptide. And this is because of a process that we will later on learn about in this course. This is because of a process called alternative splicing. So we will learn about this in details, inshallah, in this chapter. So alternative splicing, it's a process that enables the formation of more than one polypeptide, different polypeptides from a single gene. Okay? So this discovery that one gene encodes more than one polypeptide makes this statement incorrect and valid. At the same time, okay, so let me repeat what I just said, because some people had some internet issues. Scientists discovered that some genes, actually many genes, they encode more than one polypeptide, which makes this statement invalid. One gene, one polypeptide. Actually, one gene can encode more than one polypeptide. Okay? And some genes, actually, they do not encode polypeptides. So a gene might actually encode an RNA molecule only, not a protein. For example, we have genes that encodes the tRNA that you learned about in Biology 101, or the ribosomal RNA, or ribosomal RNA, okay? So as you can see now, this statement is not valid because some genes, they encode more than one polypeptide, and some genes, they do not encode polypeptides or proteins, they encode RNA molecules. Okay? So now, the last version of this statement is revised, and now we could say that one gene could encode product or products. And this product could be a polypeptide, or it could be just an RNA molecule. So now we have gene products rather than proteins or polypeptides. Okay? So gene, let me add here, gene products. One gene, gene products. So this is the right statements that describes the relationship between genes and, let's say, proteins or gene products, the products that, and these products, the polypeptides, or whether they are RNA, they are responsible, they will have a function, okay, and will be responsible for certain phenotypes inside the cell. Okay? All right, so now we should start learning about the basic principles of transcription and translation. Remember, transcription and translation both are processes or stages of what we call gene expression, التعبير الجيني. So RNA is the link between genes and proteins. You have to understand, genes are made of DNA, they are made of nucleotide. Genes, they have a specific sequence of nucleotide. And this specific sequence of nucleotide contains information important for the synthesis of proteins with the specific sequence of amino acids. And that specific sequence of amino acids determines the structure and the function of the proteins. And you've learned about this in Biology 1, Chapter 5. Now, in order to understand the information stored in genes and translate that information into proteins, two processes must happen: transcription, نسخ, and translation, ترجمة. And remember that when we talk about transcription, نسخ, translation, ترجمة, these two words are used in the context of whenever we talk about languages. لغات. When we talk about languages, we use transcription, translation. So transcription, نسخ, you transcribe something in Arabic. Translation, you are translating from Arabic to English or English to Arabic or from one language to another language. So we could, let's say, explain the process of gene expression in linguistic terms, or we could use the, let's say, we could use, we could explain these two processes by, as an example of, transcription in a language, or whenever we translate one language into another. Okay. So you have DNA, which is, this is basically the flow of information that happens. Of course, proteins or RNA, it doesn't really matter. But this is the flow of information, okay, inside our cells. Flow of information. We have information, some information stored in DNA. This information is transcribed. Okay, so we have transcription, and then we have translation. So transcription or نسخ, this is when you write, this is when you copy some text in a language in the same language. So if I want to transcribe this sentence in English, I will transcribe it the same thing, but using my own handwriting. So I would say RNA is the bridge between blah, blah, blah to the end of the sentence. This is called transcription, نسخ. Because here, in linguistic terms, we have two types of languages, let's say. We have proteins, which is written, which could be considered a language, written in the letters of, let's say, amino acids. We could consider amino acids. So we have, let's say, 20 amino acids. Imagine these are the letters of this language that is important for the synthesis of proteins. We have DNA and RNA. They are both made of nucleotides, 4 nucleotides, and 4 nucleotides. just like any language, different languages, they have different number and different types of letters. So you could think of DNA and RNA as being the same language written using these 4 nucleotides. Of course, here we have, let's say, A, C, G, and T letters. Here we have the same thing, A, C, and G, except instead of T, U. You could say that T equals U. Okay? And here we have another language that is used to make proteins made of 20 amino acids or 20 different letters. Okay? So when the information is passed from DNA to RNA, this is called transcription, because simply you are rewriting the information in the same language, in the same letters, nucleotides. But when you convert the information from RNA to proteins, this is called translation, because here we have a different language written in different letters. Here we have another language written in different letters. Okay? So this is translation. This is like when I rewrite this sentence with my hand, this is called transcription. But if I want to convert this sentence in the English language into an Arabic language, this is called translation. Same thing applies to gene expression. We have transcription, called transcription because you are writing the information stored in DNA into information stored in RNA using the same language. This is why it's called transcription. Then you are writing the information stored in RNA into information stored in proteins in a different language, or using different letters, or using different monomers. That's why it's called translation. So between DNA and mRNA, we have proteins, we have a molecule called mRNA, the messenger mRNA. Messenger means Rasul, which means this is a molecule that will carry the information from DNA into the ribosomes, okay, in order for the ribosomes to translate that information and use it to synthesize proteins. Okay. So in the next couple of slides, we will look at the differences in translation and transcription in prokaryotes and eukaryotes. Let me stop here and we will take 5 minutes break. So now let's have just a quick look at the differences in how transcription and translation happens in eukaryotic and prokaryotic cells. So to the right, we have typical eukaryotic cells. And to the left, we have typical prokaryotic bacterial cells. So we know that, from what you learned in Biology 101, Chapter 6, eukaryotes, they have membrane-bound organelles, and they have their DNA enclosed inside a nucleus, and the nucleus basically will have a nuclear envelope. So the DNA will be inside the nucleus. Prokaryotes, like bacterial cells, they do not have, let's say, membrane-bound organelles, and their DNA is just in the cytoplasm. This is why transcription and translation in these two different cells happens in different locations. So transcription happens inside the nucleus in eukaryotic cells, and translation in the cytoplasm. In bacteria and prokaryotic cells, both transcription and translation happens in the cytoplasm. And in prokaryotes, DNA is transcribed into mRNA, and then mRNA is translated into a polypeptide using the ribosome. Okay. In eukaryotic cells, we have transcription happening inside the nucleus, and the DNA is converted, the information DNA is transcribed into information in the form of mRNA, and this first mRNA is called pre-mRNA. This pre-mRNA must be converted into an mRNA, so this is a pre-mRNA, or sometimes it is called primary mRNA. This mRNA will not be translated through a process called RNA processing, which we will be learning about this process in this chapter. The primary mRNA or pre-mRNA is converted to mRNA, and now this mRNA can leave the nucleus through the nuclear course in the nuclear envelope, and now it can be translated by the ribosome in the cytoplasm. Okay? So there is an extra step between transcription and translation, which is called RNA processing, that happens inside the nucleus of eukaryotic cells. Okay? So this is how the genetic information flows. So if we have from DNA to RNA to protein. DNA is, the information DNA is converted into formation of RNA by a process called transcription. That's it. And translation using the ribosome, will be used to build or to synthesize proteins. We have transcription is carried out by an enzyme or a protein called RNA polymerase. Okay. In Biology 101, you learned about how DNA could be used to make DNA in a process called DNA replication. Okay? And DNA replication requires an enzyme called DNA polymerase. So the DNA polymerase uses DNA, single-stranded DNA, as a template to make more DNA. The RNA polymerase uses DNA as a template to make RNA molecule. Okay. Some enzymes can convert RNA into DNA. Okay, and this process is called. Let me use this color. Reverse transcription. So it's the reversal of the more common type transcription going from DNA to RNA. This is called the reverse transcription. And it is carried out by an enzyme called reverse transcriptase enzyme. We will learn about this process and the enzyme in other chapters. Okay. So now we're going to start learning about the details of transcription and translation. But first, we have to look first at the genetic code. Okay, remember, DNA consists of nucleotides. DNA codes for information or stores information that is necessary for the synthesis of proteins inside the cell. the function of the transcription translation processes and their proteins like the RNA polymerase and their ribosomes is, so there's a code here. There is a code stored in the DNA, and transcription and translation will basically decode this code in order to synthesize proteins. Code شفرة وراثية. decode فك الشفرة in order, or code, or maybe information, معلومات. Okay, as this code in the DNA is simply a sequence of nucleotides. So this is, it's not like a code. It's not a hidden code. It's a sequence of a specific sequence of nucleotides. And like A, T, C, G, 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 C, T, C, T, T, blah, 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 blah. This is just a code, okay? So we're gonna learn how these codes are decoded, or the information in the DNA, or the instructions in the DNA, instructions, تعليمات. So code, information, معلومات, instructions, تعليمات. Sorry. Sometimes the space. Instructions. Codes, information. It's the same thing. We have instructions written in the language of DNA, and these instructions must be transcribed first and translated by the ribosome, and the ribosome uses these instructions to build the proteins. Okay? Just like when you have something, you buy something new, and this something has basically a manual or a user guide that will help you assemble this something, or tells you how to use it or how to build it. Okay. Of course, going from DNA or reading the instruction of DNA and RNA to build proteins requires a translation and transcription. We have a specific language that is used to write the instructions of DNA and RNA, and this language consists of 4 letters or 4 nucleotides, but the language used to write proteins consists of 20 amino acids. So there are 20 amino acids, but there are only 4 nucleotides. So basically, one nucleotide does not equal 1 amino acid. Why? Because we have 20 amino acids. So we need 20 nucleotides, and this is not true, because only we have 4 nucleotides. But if we have 2 nucleotides for every amino acid, so 2 nucleotides, which means we have 4 different options for the first nucleotide multiplied by 4 different options for the second nucleotide. So this is the first nucleotide, this is the second nucleotide. In this case, we have 16 different. So if each amino acid requires 2 nucleotides, we will have only 16 different amino acids. And this is still not enough to encode the 20 amino acids. So 2 nucleotides will not specify or encode 1 amino acid. But if we have 3 nucleotides, they might, every 3 nucleotide might specify 1 amino acid. Because 4 by 4 by 4 equals 64, which is more than the 20 amino acids that we know that are used for building proteins. So the genetic code consists of 3 nucleotides, okay, that are grouped together in order to specify a sequence of amino acids. So the genetic code is a triplet code. Triplet means 3. So every 3 nucleotides, they represent a triplet code. Each triplet code will specify 1 amino acid. So we could look at the language that is used to write the instructions or the information necessary to build proteins in the DNA as a language made of words, and all these words are made of, each of these words are made of 3 nucleotides. So the triplet code is a series, مجموعة, of non-overlapping. Non-overlapping means لا يوجد تداخل بينهم. 3 nucleotide words, okay? And then these words of the gene, they are transcribed into complementary non-overlapping 3 nucleotide words of mRNA. Then these non-overlapping 3 nucleotide words of mRNA, they are translated into a chain of amino acids. Okay? Each 3 nucleotide word will specify 1 amino acid. Now, this figure shows how the triplet code or the genetic code in DNA is converted into a genetic code in the mRNA in the form of mRNA. Then the mRNA is translated into a code made of amino acids to form a polypeptide. Imagine this is a gene. First of all, DNA is double-stranded, mRNA is single-stranded. Now, in the DNA, we have 2 strands. One of these strands will always be used as a template by the RNA polymerase. Remember, we have an enzyme called RNA. You learned about DNA polymerase in Biology 101 and how the DNA polymerase can make more DNA or can replicate DNA or duplicate the DNA. Here, we have a similar enzyme with a similar activity, but instead of using DNA template to build more DNA, this enzyme uses a DNA template to make an mRNA molecule or an RNA molecule. So one of the strands will be called the template strand, okay? Because its sequence will be used by the RNA polymerase to build a complementary mRNA strand. So as you can see, this strand is complementary to this strand. So here we have A. It should be T, but since we are talking about mRNA, instead of using T, the enzyme uses U. C, G, C, G, A, U, U, C, C, G, 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 C, A, T, U, G, C, T, A. So imagine this is a gene, this is the mRNA. Now, the genetic code of the triplet code of this DNA will be something like this. This is the first code made of 3 nucleotides. This is the second code or codon, and this is the fourth one. So this is the first one, codon 1, this is codon 2, this is codon 3, this is codon 4. Each codon or each code consists of 3 nucleotides. These 4 codons, by the process of transcription and by the activity of the RNA polymerase, are converted into codons or codes in the form of mRNA molecule. Now, the mRNA molecule is single-stranded DNA molecule, single-stranded nucleic acid molecule. Then, by the action of the ribosome and many other molecules during translation, these codons will be translated into amino acids. These codons will be used by the ribosome to build a polypeptide. This is a polypeptide. It's more accurate to call it a polypeptide, not a protein, with a specific amino acid sequence, with a specific order of amino acids. Now, each codon specifies one single amino acid. So each time UGG will be read by the ribosome, the ribosome will understand that this codon means tryptophan. Each time the ribosome reads UUU, it will understand that the ribosome, this codon, indicates or suggests or specifies the amino acid phenylalanine. GGC, glycine, UCA, serine, and there are many other codons for the different 20 amino acids. Okay? Now, this strand is called the template strand because it will be used by the RNA polymerase as a template, which means the other strand is also called, we could call this the non-template strand. Another name for this, the template strand, is the non-coding strand, which means this is strand, the non-template strand is called the coding strand. Notice that the sequence of the non-template strand or the coding strand is similar to the sequence of the mRNA. You just need to replace the T with the U. So here, TU, GG, GG, TU, UU, GG, C, TU. It's called coding strand because this one is actually contains the code that will be converted into a code in the mRNA form. Okay? Another name for, so we have 2 names so far for the 2 different strands. The top one here in this example is called template strand or non-coding strand. The one at the bottom here, it's called the non-template strand or the coding strand. Also, this one could be called the sense strand. And this one, the top one, is called antisense strand. Sense means mantiqi, because this is the strand that makes sense, because its sequence makes sense, and its sequence will contain the codons that make sense, and it will be realized by the ribosome to build the polypeptide. Okay? So this could be called the non-template strand, coding strand, sensitive strand, or transcribed strand. Sorry. Non-transcribed strand. Sorry. Go back. Remove this. Okay. Or the ones that is used as a template could be called template strand, non-coding strand, or anti-sense strand. All right. So a particular gene could be transcribed from the top strand or the bottom strand, but always from the same strand. So basically, if you have here like gene 1, gene 2, gene 3, each gene consists of 2 strands. So maybe the direction of transcription will be like this from the bottom strand, this from, in our example here, so if, so this part is basically, and notice that the RNA polymerase synthesizes the mRNA from the 5' to 3' direction. Okay? So this will be used as a template, and the polymerase will start adding 5, the first nucleotide in the direction from 5' to 3. Now, if we zoom in into this gene, let's say, now we have 2 strands, okay? Maybe something like this. If we zoom in, we have 3', 5', 3', 5'. Similarly, we have here, if we zoom in to this region, we have 3', let's say 5', 3', 5'. So in this example, the blue gene, the top strand was used as a template. But maybe for the green gene, maybe it's the bottom strand. So maybe it's not the top strand that is template, but maybe this is the template strand. For the yellow one, maybe it's the top strand is the template strand. So for each gene, which strand will be used as a template one, which one is the coding, it depends on the gene, and it depends on some sequences before the gene that will determine which, before or after the gene that will determine which strand will be used as a template, which strand will be used as a coding. But each time, if each time the polymerase binds to this gene, it will always use the same strand as a template strand. So each time the polymerase binds to gene 1, it will use this strand as a template strand. It will always use the bottom strand as a template strand. Here, it will always use the top strand as the template strand. Here, it will use this strand always as the template strand. Okay? So the template strand is always the same strand for a given gene. But different genes, they might have different template strands. It could be the top or the bottom. In this case, for gene 3, it's the top. For gene 2, it's the top. For gene 3, it's the bottom strand. Is it clear? So whether the top or the bottom template will be strand will be used as template depends on the orientation, how the polymerase will bind to the gene or to sequences within the gene. Okay. During translation, the mRNA will be read. The mRNA will be read by the ribosome. The ribosome reads the mRNA in the 5' 3' direction to synthesize the polypeptide in the from the N terminus to the C terminus direction. Okay. All right. So remember that we have 4 kinds of nucleotides in the DNA. And each 3 nucleotides, they determine 1 amino acid. So we have the first nucleotide plus second nucleotide plus third nucleotide. All of these will give us only 1 amino acid. So the first nucleotide could be 1 out of 4. So there are 4 different options. 1, 2, 3, 4. It could be A, it could be C, G, or T. The second nucleotide could be also 1 of 4. It could be A, C, G, T. The third nucleotide, it could be also 1 of these 4 nucleotides. So if we multiply 4 by 4 by 4, we have 46 different potential codons or triplet code. This is the triplet. So scientists, after discovering that each amino acid is specified or assigned by 3 nucleotides, scientists determined the different 60 codons or 64 different triplet codons or triplet codes, and they wanted to know which codes or which codons or which specific sequence of 3 nucleotides will specify which type of amino acids. So this table lists the 64 potential codons or triplet codes and what type of amino acids they specify. So as you can see, more than 1 codon or more than 1 triplet code could be specifying the same amino acid. So the amino acid leucine is specified by 4 different codons. Choline, 4 different codons. Histidine, 2 different codons. Glutamine, 2 different codons. Threonine, 2 different codons. Okay. One of the codons, AUG, is called the start codon, which also specify the amino acid methionine. And 3 codons, they do not specify an amino acid. They are called stop codons. Okay. We will learn about these codons, inshallah, later on, maybe tomorrow. So let me stop.